Hey A&Pers, this is Dr. Jones checking in. This is A&P 1, Lab 1, video 6 maybe. We're going to 6. Sure, why not? Okay, so we need to talk about mitosis, which is not just what you say when your sister steps on your foot. Mitosis? I know. I warned you, didn't I? First video? Yeah, I think I warned you. You've been warned now. Okay, so mitosis is a process of cell division. So there are two types of cells, and those are somatic cells, which are body cells, and sex cells, which are the gametes. There are two types of gametes. In layman's terms, they are eggs and sperm, which is oocytes and sperm. Yeah, sperm is a technical term. Okay, so the cell division that makes sex cells is called meiosis. And that's the cell division that makes gametes. And we will talk about meiosis in great detail at the very end of A&P 2. But for today, we're talking about mitosis. Mitosis is the cell division that makes somatic cells. And somatic cells, soma means body. So somatic cells are all the cells of your body that are not an egg or a sperm. So all of it. Okay, so the phases of the life cycle. The first phase is interphase. An interphase is when the cell is not undergoing mitosis. When the cell is not dividing. There we go. And then there are the phases of mitosis. And the phases of mitosis go in order. Prophase, Metaphase, Anaphase, and Telophase. Now, one of our books recommends that you think of the little oddly shaped fluffy mat that goes in front of your toilet and think of it as the pee mat. How about no? I taught my son to not pee on the floor. I know, it was difficult, can be done, takes time, give it time. Keep reminding them. It's like washing their hands with soap. You just gotta keep telling them. So I like to include the I of interphase, which is not a phase of mitosis, and make a mnemonic for the entire life cycle, which looks like I pass my anatomy test. That one's my preferred. But be aware that interphase is not a phase of mitosis. It's a part of the cell life cycle when the cell is not undergoing mitosis or meiosis. We'll talk about meiosis next semester. So let's talk about this model, which is the model for interphase. Now on the previous model, you had cell model. Look. The centrioles were the Christmas candies, the little Twizzler nibs off to the side. There's always two of them, and they're always at right angles to each other. The area they're in is called the centrosome. Again, some means body, just like soma means body. Okay, so centrosome. On this model, the little two black dots are your centrioles, and this area around it is the centrosome. Okay, so this is interphase. You have the nuclear envelope, you have the diffuse chromatin in a non-dividing cell. Chromatin in a non-dividing cell. And as a reminder, what the chromatin looks like, this will be the red fuzzy stuff on the cell model. In prophase, and both of these models represent prophase. This will be early prophase, this will be late prophase. For my purposes, you don't need to know early versus late prophase. You just need to put prophase on the test for either one. Several things happen in prophase. The nuclear envelope dissolves. Which has happened. Here you have a nuclear envelope, here you don't. The centrioles separate. And as a note, since y'all are college educated now, separate, it has a rat in it. S-E-P-A-R-A-T-E, separate. Centrioles separate and form the mitotic spindle. Which is also called the spindle fibers. Okay, so here you see these centrioles separating and you're starting to form the mitotic spindle. Here you can see the centrioles, which again on these models are the black dots, have separated, and each of these red lines is a spindle fiber. The other thing that happens is the chromatin coils, and then it coils, and then it supercoils. Maybe 2 steps, I don't remember. The chromatin supercoils into visible chromosomes. Visible. Oh, you can't even see that. Sorry. I'll zoom in. There you go. Visible. Also, I haven't yet apologized for my handwriting, so this is your apology for my handwriting. Chromosomes. Okay, now you can see the whole spiel. So, interphase, we have a nucleus, we have centrioles. Perfect. Okay, so prophase, the nuclear envelope dissolves, the chromatin coils into visible chromosomes. So what happens, it's just like if you take a shoelace and you coil it up, coil, 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 and you just keep twisting it the same direction until it makes like a spiral that it stays in. And that is your supercoil. And the chromatin also coils around some proteins. It makes, we'll do all that in lecture though. That's part of lecture, we'll get there. Okay, and I call them visible chromosomes, where they're called visible chromosomes, because this chromatin is so small and diffuse that it is not visible on a light microscope. The light microscopes are the ones we will use in lab if we're having lab in person. These visible chromosomes are supercoiled chromatin, and they're so much thicker that they are visible on a light microscope. So that's why they're called visible chromosomes. They're not visible without the naked eye, but they are visible with a light microscope as opposed to like an electron scanning microscope or something like that. And then we need a picture for prophase. Here we go. Prophase, and then we've got the centrioles at the opposite. And then we've got visible chromosome squiggles in the middle. So here's our interphase picture. You've got a nucleus and the 2 centrioles together, moving. And prophase, the centrioles are separated. In fact, let's add some spindle fibers. There we go. Looks right now. Oh, I went out of screen. There you go, spindle fibers. Okay. And now for metaphase. Metaphase, I'm going to zoom back out a little bit so you can see the whole page. Chromosomes line up in the middle. Chromosomes line up in the middle. Okay, and here's our model for metaphase. So you can see they line up, these gray squiggles are the chromosomes, and they line up in the middle. And the plate that they line up on is called the metaphase plate. On metaphase plate. There you go. And of course you still have the black dot, which is the centriole, and the red line is a spindle fiber. And the gray squiggle is the chromosome, and the plane that they're on, pink here, is the metaphase plate. So, name the phase of mitosis, metaphase. Name the phase of mitosis, prophase. Name the part of the cell life cycle. Interphase. Remember, I won't ask phase of mitosis for interphase because this is when the cell is not undergoing mitosis. Okay. And then anaphase, the chromosomes are being pulled apart. Apart, off screen again. Anaphase, apart, metaphase, middle. Get it? Okay. So, oh, we didn't have a picture here. Chromosomes are lined up in the middle. And what happens here is each chromosome is attached to the centriole by a spindle fiber. Okay. So then in anaphase, the spindle fibers shorten, and that pulls the chromosomes apart. Okay, so here are your two models of anaphase. Now this is early anaphase and this one is late anaphase, but again, for my purposes, these are both called anaphase. You can see that the chromosomes are being pulled apart, and in fact, these top ones, if they had little crossbars, would be a capital A. So that's how you can tell anaphase. And on this one too, makes a capital A. They're being pulled apart. Now, sometimes, especially on the slides, it's hard to tell the prophase from the anaphase. But remember, in prophase, you'll have just a rando ball of squiggles in the center of the cell. Whereas in anaphase, you have these very neat A's and V's being pulled apart. Okay, anaphase. Now, I'm going to draw another picture of anaphase and address the difference between our two anaphase models. And it's that right there, which is called the cleavage furrow. The cleavage furrow is an indentation that indicates that a process has begun, and that process is cytokinesis. Am I off screen again? Okay. Cytokinesis, so mitosis is the division of the genetic material between the two daughter cells. B/W means between. Two daughter cells. Cytokinesis is the division of the cytoplasm That's an M. I swear it's an M. Between two daughter cells. Cytokinesis begins in anaphase. Continues through telophase. I'm off screen again. There you go. So mitosis is the prophase, metaphase, anaphase, and telophase, the division of the genetic material between the two daughter cells. Cytokinesis begins here in the middle of anaphase and continues throughout telophase, and it divides the cytoplasm between two daughter cells. Begins in anaphase, continues through telophase. You see all that? Yeah? In focus and all that good stuff. Excellent. So, name the phase of mitosis, anaphase. So, no cytokinesis, cytokinesis. And what's the indentation called? Not a butt crack, a cleavage furrow. Cleavage furrow. And more importantly, the cleavage furrow indicates that what process has begun? Cytokinesis. And then telophase. And you continue to have cytokinesis. You can see that compared to the anaphase, the cytokinesis has continued. And telophase is the opposite of prophase. We will now undo everything prophase did. So the nuclear envelopes reform. The chromosomes uncoil into chromatin. Chromatin. Chromosomes uncoil into chromatin. Let's see, what else do we have? Well, the mitotic spindle will dissolve, but okay. And then once you have cell membranes, two cell membranes here completely separating the two cells, you have two daughter cells. Now here's the kicker, because I skipped some stuff. The kicker is each daughter cell, new page, each daughter cell has the same number of chromosomes as the parent cell. Each daughter cell has the same number of chromosomes as the parent cell. How is that possible? Well, I skipped a set. Right at the end of interphase, move these models. Right at the end of interphase, before mitosis begins, is a step, this is during interphase, called DNA replication. How many chromosomes do humans have? The answer is 23 pairs. And if you have 23 pairs of shoes, how many total shoes do you have? So 46 total chromosomes. We'll talk more about that at the end of A&P 2. But for now, DNA replication takes your 23 pairs of chromosomes, 46 total, and multiplies it by 2, so that you have two, hopefully, identical copies of your 46 total chromosomes. And during prophase, those are all squiggled in the middle, and one set become attached by the spindle fibers to one centriole, and the other set of identical copies become attached via spindle fibers to the other centriole. So when the chromosomes are lined up in the middle at metaphase, the top are one copy of DNA and the bottom are the other copy of DNA. And then during anaphase, one copy is pulled towards one centriole, the other copy is pulled towards the other centriole, so that during telophase, and then further when it's two daughter cells, each daughter cell ends up with one entire copy of the genome, which is the 23 pairs of chromosomes. So each of these daughter cells has identical chromosomes. In fact, every cell that has a nucleus, every nucleus contains all of the genes needed to make your entire body. And we have officially doubled our 10-minute time limit that I've given myself. But this is both lecture and lab material you need to know about mitosis. So this has been Dr. Jones, A&P 1, Lab 1, Video 6. Thank you as always for tuning in. The education must go on. Dr. J out.